Hello and welcome back to all. Today we are discussing the topic of gene editing. Recently, a research team from USA has developed and safely delivered a personalized gene editing therapy to treat an infant with the life-threatening incurable genetic disease. The infant was diagnosed with the genetic metabolic disorder called carbamyl phosphate synthesis 1 CPS1 deficiency. Its deficiency can cause to build a large amount of ammonium acid. It's a chain of protein. Because of this the protein can lose their efficiency to fully break down byproducts, resultant severe damage to our brain and liver. Treatment like liver transplant is mitigate these diseases, but treatment for minor child is a low protein diet. The team of researchers customized the therapy using the gene editing platform CRISPR, which allowed corrections of a specific gene mutation in the baby's liver cells that led to the disorder. Gene editing is the technology that can change DNA sequence at one or more points in the strand. It also involves removing or changing a single base or insert a new gene along together. The process involves three major steps that include insertion, deletions, and modification of the gene of interest. Types of gene editing: somatic gene editing and germline gene editing. Here, simply we can say somatic genome editing is it shouldn't directly target on our reproduction cells, but germline genome editing is directly targeting our reproductive cells. It refers to the alteration of cells that cannot contribute to gamete formations and this cannot be passed on from the individual to offspring. Targeted cells, non-reproductive cells like skin, liver, kidney, muscle cells. Germline genome editing is genetic modifications of reproductive cells or embryos that creates changes which can potentially be passed down to future generation. Targeted cells, reproductive cells like germ cells, embryos or gamete productive cells. Challenges associated with gene editing. Ethical concerns, embryo research ethics. Many people hold deep moral or religious convictions that human embryos represent human life that deserve protection. From this perspective, using embryos for research purpose, which may involve manipulating, experimenting on, or ultimately destroying them, is ethically problematic or even morally unacceptable. Informed consent. There is the debate about whether truly informed consent is possible for germline therapy, since the patients affect the embryos and future generation who cannot consent for themselves. Justice and equality. Like many new technologies, there's concern that genome editing will only be accessible to wealthy individuals, potentially increasing healthcare disparities. Safety concerns, safety issues. The primary concerns centers on off-targeted effects, edit occurring in unintended locations and where only some cells carry the genetic edit while others don't. Due to this risk, researchers and ethicists generally agree that germline genome editing should not be used clinically for reproductive purpose until it's proven safe. Regulatory inconsistency. Experts fear that varying global regulations on gene editing could lead to controversial use like designer babies. Gene editing technologies offer a revolutionary potential to treat incurable genetic diseases, improve agriculture, and address environmental challenges. However, realizing this promise requires careful navigation of ethical concerns around safety, equality, and consent, particularly for germline editing that could affect future generations. Thank you.